I will present today a study about a potential group of mimicry. I will show evidence about this new mimicry system, and I will discuss some cool questions that can be investigated in this. So, axiomatic species are species that are toxic or unprofitable for prey, and they somehow advertise that condition to predators by having warning coloration. And it is often registered that there is great variation in color morphs in this species. It is not very well known, though, how this variation arises, but once it arises, it can be maintained by positive frequency-dependent selection. For example, if you have two different color morphs separated geographically, migrants between these two color morphs will be strongly selected against by predators because they are rare and those predators are not familiarized with those. So some other interesting pattern is that when you have multiple axiomatic species co-occur in the same area, they can converge to the same axiomatic signal. And this is a phenomenon known as Müllerian mimicry, and it has been observed in many taxonomic groups, as you can see here, frogs, millipedes, birds, and many others. And this happens because it is convenient to converge to the same axiomatic signal because somehow the two species share the cost of training predators about their and the result of this is that you will have a remarkable matching of color morphs between the different species, and also usually extreme diversity in this group. So you can see these famous examples of heliconius, butterflies, and some other butterflies from the however, also, although mimicry can be a trigger for high phenotypic diversity in these groups, in axiomatic species, mimicry actually is not required, and you can see high phenotypic diversity in axiomatic species without mimicry, like, for example, in this Munoz-Ramirez, poison frogs from Panama. Now I will introduce this group of beetles. These are ground beetles from the temperate forest of South America. They have a number of species, and all the species are very diverse, and this diversity is geographically structured. They are relatively big, about 2 to 3 centimeters. They don't fly, they are wingless, but they usually present this conspicuous coloration, which is metallic and bright. And they also have chemical defense, which is a characteristic of the entire family. But they are very well known where they live because they are very pungent. Another characteristic is that at least 3 species, the 3 main lineages of this group, are widely co-distributed in Chile. And more interestingly, there seems to be a pattern of covariation in phenotype between different species. So you can see here some areas like this, here. There are the 3 species, they are the same 3 species. The 3 species seem to converge to the same axiomatic signal. But this pattern is not very clear anywhere else, because you can see also that some other areas this doesn't hold, and you can see more diversity in axiomatic signals. So now the question is, do we see here really a case of Müllerian mimicry, or do we see statistical support for color convergence between these 3 species? We want to answer this question by first quantifying coloration and then testing whether co-distributed species are more similar than expected. Given that these species are highly polymorphic, it seems that that chance could be a possibility, and just happens to, some locality happens to have morphs that are similar in color. So we collected data, color data with from about 200 individuals from these 3 different species and across most of the distribution. And we also collect data to build a phylogenetic analysis. Basically, we projected the color data in a way that it's seen by predators and potentially avian predators. Avian predators here are the ones that can see, actually discriminate these different colors. And then with this data collected, we mapped every population of species in a color space. Then the question that we want to ask is very simple. We calculated all the distances between populations, but all distances interspecific distances between species. And we asked whether, or we compared within the same locality between species versus distances between localities. And we compared the frequencies. So we found that distances between species, but within the same locality or the same population, are, well, on average, lower, much lower than distances between sites. And these differences are statistically significant based on randomization. Well, in addition to that, phylogenetic analysis showed that well, color is not explained, this color similarity is explained by shared ancestry. So you don't see like color being clustered by species. And you can see that different colors arise several times independently within each. So summarizing, we have that these species present overlapping distribution very widely, and they showed conspicuous coloration, chemical defenses, and now show that there is color similarity or matching that is significant. So all these properties make a very strong case for Müllerian mimicry. The question now that we have and it arises is how then we can explain those areas where mimicry seems not to be evolving. There are many reasons. Here I include a few of them, try to discuss potential. And for example, one possibility is that just selection is not the same everywhere. There are areas maybe where selection is strong, mimicry, while in other selection is weak, perhaps. And this might be due to differences in predator communities, or maybe there are same predators everywhere, but they behave differently. Unfortunately, there is not much ecological data about diet of birds or study cases where document these beetles are being predated by birds, if they are being predated, how. Well, another possibility is that maybe selection is the same everywhere, but maybe there are areas with higher gene flow and others low gene flow, so that maybe high gene flow is overrating selection. So maladaptive genes can migrate between areas, and so it's maintaining more diversity. However, this possibility might seem unlikely in this area because the forests are very continuous everywhere. There are no very clear differences in climate or geographic barriers. And despite that, this continuous landscape, you still see geographic morphs very closely separated, and they still maintain the And another possibility is that maybe some areas species have not coexisted long enough for evolution to try this pattern. Maybe so what we are seeing maybe is that evolution is in progress, so we are seeing an intermediate step toward convergence. This certainly is a possibility, especially in South Chile, because we know that glaciation really greatly impacts South Chile. And based on our data, we see that there is less convergence patterns through the south, from north to south. So now, just want to highlight that this system is very remarkable in terms of showing this geographic pattern variation in mimicry. And I think it can help to really ask very interesting questions about evolution of mimicry, how it is evolved, how it is repeated. In that, I will just thank my supporters and Munoz-Ramirez for all their no, and unfortunately there is no data on the venom, and if it's the same venom in all the species, or maybe in guards, or in guards space. Some of the things. But because this is a characteristic of the entire family, and all these species are very well known, most likely a similarity.
Just a technical question with the spectrophotometry of the color. Given that a lot of the coloration in insects and crabs in particular is structural and is dependent upon the angle, how does one actually calculate the color of any given spot, since it might vary?
Yeah, actually we calculated color in these beetles in a bunch of different angles. So we just keep consistency, and we show results from a particular angle. We have seen this pattern across all the
I wasn't questioning the results, I was just more curious about
yeah.
measure it. Yeah. Thank you.
Are the colors of the
Yeah, no, the color is not based, it's structural, based on the physical structure. Which makes